Hello, and thank you for joining us today. Our webinar today is Single Cell Analysis with the MVX 7100 Workstation. Our presenter is Peter Winship, Product Manager for Teledyne CETAC. Okay, I'm going to go ahead and turn it over to you, Pete.
Lovely, thank you very much. Thank you, Shelley, for the kind introduction. Hello, everybody, and thank you for joining. So as Shelley said, my name is Pete Winship. I'm a product manager at Teledyne CETAC Technologies, and one of the systems that I'm responsible for is the MVX 7100 microliter workstation, which I'll be talking about today in conjunction with single cell analysis, which is a very new, interesting, emerging type of analysis, which there's a lot of interest in currently. So before I move on with the actual main presentation, I'll just go through a quick agenda, just to go through what I'm going to be talking about today. I'll give a very, very brief introduction to cell analysis with ICPMS and just where that's come from as a concept. Then I'll be going through what people refer to as this single cell analysis, the single cell analysis. What is it? What does it mean for analytical chemists in ICP laboratories around the world in terms of biological analysis, etc. After that, I'll go through basically a workflow of sorts, starting with a sample, where that might come from, what that might mean to the analyst in terms of how it needs to be looked after and introduced, all the way through to generating data. And then following that, which is going to be most of the presentation, I'll then go through some sort of wider analytical considerations, which I suppose I'll be throwing these out into the ether for ongoing work and discussion. So cell analysis and ICPMS. Really, I'm just going to bring up one slide for this. ICPMS technology has been developing at a remarkable rate, and certainly over the last 20 years that I've been involved with it, and there's a lot of people who've been involved with it for considerably longer than I have. In that 20 years, there's been a huge development and shift in the capability of ICPMS. You know, the ability to be far more sensitive, to be faster, to have systems that are far more robust to sample matrices. And as part of that process, obviously, the possible applications have grown and developed. The pushes in sensitivity have allowed analysis and detection, which wasn't possible in previous years, and now is very straightforward. And this development has taken us to a point that started, I don't know, maybe 5 to 7 years ago, where we were far more confident in detecting what you might refer to as single events. So the detection of nanoparticles and microparticles, and beyond that, what we're going to talk about today, single cells. So with the ability to actually detect individual cells coming through a plasma, there has been a lot of focus put on this in terms of research and analysis on the elemental composition of different cell types. So, you know, given that biological cells have metals associated with them in the simple ways that you might expect, phosphorus, zinc, copper, etc. So there is a elemental content interest in individual cells in what you might refer to as their natural state. There is interest in cells in terms of cellular uptake, so how a cell might take up certain what you might refer to as nutrient elements, but as well as that, contaminant elements. And then one of the main areas of interest is the cells and their uptake of metal-based drug compounds or nanoparticles. So probably the most well-known of which is platinum-based anti-cancer drugs such as cisplatin and oxaliplatin, and how on passing into cells that they can then bind with and destruct DNA. So there's numerous areas of interest in terms of cells and the elemental composition, and this is now a huge area of research, which I won't dwell on now from here, but that's in a nutshell the interest in cells in terms of ICPMS. So this term single cell analysis is mentioned a lot, and what does that actually mean? So just to, what I want to do here is I just want to compare what a single cell analysis is, or a single event analysis is, in relation to what you might refer to as a typical sample introduction. So normally for liquid sample introduction, you will have a dissolved analyte in some sort of solution, some sort of media, and a certain volume as well. Now that sample, that solution, will be continuously pumped, introduced somehow, whether that be with a peristaltic pump or with a syringe pump, continuously pumped into the plasma of an ICPMS. And what you'll have is that analyte, those molecules that you're interested in, those compounds, then get broken down into their constituent atoms in a plasma because of the temperature and the electrical environment that's there. Those atoms are ionized, and those ions are then passed on into an the mass spectrometer aspect of the ICPMS for detection and measurement. And what you end up with is, with a continuous introduction, you get a nice continuous signal from which you take your measurements over a certain time period from that continual signal. Now, in the case of a single event, or in this case a single cell analysis, it's a slightly different scenario. You have a suspension of cells, say, in a certain media, a certain matrix, and as those cells pass into the plasma, that cell will break down. It will be broken down to its constituent atoms again and ionized, and you end up with an ion plume. So it's a finite event, you might say. It's a small event rather than a continuous signal. What you're going to get is you'll get a spike, a peak in your mass spectrum from a particular cell, and that's what we're looking to capture the data for and take measurements on. And that is what is possible now, that we can actually detect these spikes, these peaks, from single events, single cells, from a sample as it passes through. So that is, in a nutshell, what we're looking to do. So from this, what you'll find, so you'll see here a similar comment as I made on the previous slide, but down here on the bottom right hand side of the screen, what you've got here is you've got some what a mass spectrum series of peaks over a period of time actually looks like. So this is signal intensity versus time, and what you're looking at here is a number of cells which have gone through an ICP plasma, an ICPMS plasma, and these spikes, these peaks, are what is passing through that plasma. So you've got here, these spikes correspond to cells as they've passed through. And from this data, what we can do here is a number of things. So this is where the concept, what people refer to as single cell analysis, actually gets quite broadens out, because we have to start asking the question, what sort of data do we want from this? So we can take these events, these spikes, and we can look at those and say, well, that's equivalent to the number of cells that are passing through, so we can get data on cell populations, how many cells are present, how many cells of the cell number that have gone through have a certain element associated with them. We can also look at the signal intensities of the events as they're passing through, and we can equate those to the mass of elements that are there. So we can start actually looking to quantify analyte elements that are present in these cells. So that's what we're looking to do. Now, what I wanted to probably dwell on the most was looking at the concept of starting from the sample and moving through to data. So there's a workflow associated with this, which will look very, very familiar to most laboratories, to most analysts, but there's more to talk about as we go through these steps. So what I was going to start with here is this idea of this analytical workflow. Now, what I didn't want as part of this this webinar, this presentation, was for me to put this slide up and to actually talk about this as something special. The idea of you start with a sample, you might have some automation, an interface, then an ICPMS system, and you generate some data. That is the workflow of every kind of ICPMS analysis. But what I did want to stress here of this this basic workflow was the idea that of the first, from between sample and ICPMS, there's actually more for us to consider before we get to the data point. So between sample and ICPMS, we actually have to look at things slightly differently. So what we want here is we want to introduce a sample, and our best data is going to come from getting as many of the cells that we're interested in that sample from A to B. So from our sample receptacle, in whatever form that is, through the automation that we're looking to use, through the interface, so what I refer to as the interface, the glassware interface, the nebulizer and spray chamber. From the analytical workflow, which as I mentioned, is the same for any normal ICPMS analysis. In this case, what we're interested in here is slightly different. So we will have a sample which contains our cells, but for our best data, or our best measurement here, what we want is to ensure that we get the maximum number of cells from our cell source, which might be a sample vial or a well plate. We want to get as many of those cells through the process, the automation, through the glassware interface, so our nebulizer and spray chamber, into our ICPMS. So we want a maximum transport efficiency from A to B. Now that's not typically the case with ICP systems, because we often send a lot of our sample to drain in the spray chamber. But in this case, what we're looking to do is more of something closer to a total consumption process. So we want to get as many of our sample cells from A to B, from our sample vial or well, through that process to ICPMS, but we also want to maintain the integrity of those cells. We want as many of them as possible to survive that process, to ensure that the data that we get is representative of intact cells, and that we don't lose cells which are broken down and then we lose that data in the baseline. So we want maximum transport efficiency whilst maintaining the cell integrity. Now, what I would say at this point is that there are a number of variables which all feed into achieving that, getting the best transport efficiency out of this and maintaining the cell integrity. So that's what I'd like to sort of start moving into from this point here. So starting with the sample, we've really got to consider, as analytical chemists, analytical scientists, we've got to consider where we start from. So I'm not a cell biologist, but if a cell biologist would hand a sample to me, I would have to start asking a number of questions like, you know, what are my samples in? Are they in well plates? Are they in vials? Are they in a certain media? Are there any conditions I need to be aware of? Do I need to maintain certain conditions to keep the cells viable? Do I have any other considerations as well, like are my cells likely to settle to the bottom of whatever they are currently in, a well plate well or a vial? Are there any other considerations that I have to ensure that what I'm taking, what I'm sampling for analysis is homogeneous and what I get is representative? So you have to start asking those questions right from the very beginning. And given that there's a huge range of cell types which have to be considered, we're looking at different sizes of cells, we're looking at different shapes, they'll have different integrities, so they'll be tolerant to different conditions, and that will be again, across that process from A to B, there will be differences in conditions between the sampling process through the introduction through a nebulizer and spray chamber for detection. So it's a number of considerations to have. So where we need to start with is pretty much an open mind in terms of what variables we're going to use, and what you'll probably end up doing as analysts, as people working with cell scientists and researchers. There will be a range of conditions that will be established for different cell types. So next, going on to the automation. So this is where our MVX 7100 microliter workstation comes in. So this is our starting point for the process. So we have our samples, which might be in well plates or in vials or tubes, and we put them on this system here. So this is a syringe-driven automation system that's going to allow us to take distinct volumes of sample for introduction to a plasma. So this system, this current iteration of it will it's possible to take up sample volumes smaller than 5 microliters and up to about 1.5 milliliters. And taking that volume of sample, we're then able to very accurately control the introduction process in terms of the flow rate. So we can push sample to an ICPMS plasma at flow rates as low as a couple of hundred nanoliters per minute, up to milliliter per minute, and beyond flow rates far beyond what you would need for any kind of nebulizer for ICPMS. So we've got full control over this, which is one of the important aspects of this process, to allow us to be able to work with different cell types to maintain that cell integrity so that we can get the best data out of this. We can also use this system for sample homogenization as well. So again, if we have cells which are more likely to settle over time, if you're going to run an analytical batch, if samples are sat for any particular length of time, there is a potential for some cell lines to settle according to gravity. So it might be that one of the variables to consider as part of the process here is to use the aspect of this system for sample homogenization. So we can withdraw a certain volume of sample out of a well plate or vial, and then we can put it back after it's been withdrawn. We can put it back into the same place to add a degree of turbulence to where the sample's coming from, so that we try to maintain a homogenized suspension of cells for us to do our analysis with. There's also the option here to have temperature control of your samples. So if this is important to a certain sample, certain cell types, for maintaining cell integrity, certainly over a period of time for a batch analysis, that is an option here. So we can go down as low as 4 degrees C and up to 40 degrees C. Then we've got the option to sample from numerous different types of sample vials and tubes, to have septum piercing from certain vials, and then we can sample from either 96 or 384 well plates at this stage. So that's our automation for this process. So those are the first 2 boxes of this process, process going from A to B, going from sample through automation, through the interface, the ICPMS, and then arriving at our data. So next, the interface. So the interface process of this, we've spent actually quite a while working on this in collaboration with Glass Expansion in Australia. So Glass Expansion we sat down and we talked through our requirements for this. What do we need to arrive at the idea of attaining maximum transport efficiency for cells through this process and maintaining cell integrity. So essentially that's our brief at the outset. We need those 2 things to arrive at successful single cell analysis. So Glass Expansion have done a lot of work arriving at a glassware interface for this, whereby when we introduce our cell samples through a nebulizer, that we focus those cells as they've come out of the nebulizer straight towards an ICPMS plasma. and that there's minimized or no recycling through the spray chamber glassware. So we're looking to have a nice nice baseline, introduce our cells from our sample, clear them through the glassware, and then go straight to washout, a quick washout straight away. So they've done a lot of gas flow modeling on this, and they arrived at the design which you see here on the right hand side. So there's two aspects to this interface. We have a high efficiency concentric nebulizer, which allows us to work with introduction flow rates up to 50 microliters per minute. So it's at the lower end of sample introduction flow rate, which the MVX system can meet. So we can deliver any of those flow rates from the hundreds of nanoliters per minute range up to that 50 level on this glassware with the MVX. We can drive anything within that range. So the nebulizer itself is a nice wide bore capillary, and the reason for that is, first of all, to ensure that when you're working with the cellular material, that you're minimizing the potential for any blockages. So you're getting a nice clear flow of these cells through the nebulizer. Also, the idea behind the wider bore was to try and reduce the potential for collisions between cells as they're going through the nebulizer, so that you're reducing the possibility for cell lysis. And at that point, if you do get cell lysis, you're going to lose everything to the baseline, rather than getting the peaks, the spikes that you're interested in. So that's the nebulizer, and you can see in the diagram on the right-hand side. So you've got the nebulizer, which is number 6 there, which with a nebulizer in gas inlet line, which is number 5 in that diagram there. Next, we have the spray chamber itself. That's number 3 on the diagram there. So this is a sort of like a bulbous type nebulizer, which the point of it is just to allow space for the expansion of nebulized droplets and then focusing of those droplets through to the end of the spray chamber. That's the reason for the shape of it. There's a drain on there with a drain line which is there, 0.4 in the diagram there. Now that is there to clear anything that doesn't go through, but the point is to try and introduce as much of your sample as possible, have that maximum transport efficiency. So that is there more as a contingency rather than anything else. Now, in addition to that, what you have is you've got an additional gas flow rates which can be introduced just above the nebulizer there. Now that is an additional gas flow which will envelop basically the nebulized plume and deliver that focusing of that plume of liquid droplets out from the nebulizer through to the end of the spray chamber. So it's been designed. They've looked at the design for this very, very carefully, and they've tried to meet our requirements for this on single cell, and they've done a very good job with this. It's the data that we've seen so far is very, very good, with transport efficiencies of 50, 60, 70% at the moment. Right. So, and then just to show you what that actually looks like outside of a diagram, these are the actual components. This is what it looks like. And you'll see on the doesn't like me going backwards. Hold on. So you'll see on the spray chamber at the bottom, you can just see on the left-hand side there, you can see the port for the nebulizer going in through the back, and then also the port above it for the additional gas flow. Right, so up to this point, what we've done is I've talked through where we start with our samples, our automation, and then our glassware interface. So before we move on to ICPMS, really I've mentioned transport efficiency and the kind of transport efficiencies that this glassware can deliver. But what about the cell integrity? So this is a very, very important aspect of this process, and we need to be sure that cells of different types with different integrities, certainly in terms of their structure, it's possible for them to pass through this without any cell lysis or minimize cell lysis. So what we have next is some really nice microscopy images which have come via way of our associates at Agilent Technologies. So what they did with this glassware and the MVX system was they looked at this cell line here. This is K562, which is a leukemia cell line. And they looked at cell sizes between 10 and 20 micrometers in size. Now these cells are very common in research. They've been looked at routinely since the 1970s. They're very easy to culture and isolate. And these cells were put through from well plates or vials through the MVX system, through the sample loop that's on the MVX system and the 6-port switching valve, through the glassware, and then they've looked at them again at the other side at different nebulizer flow rates to see what the survival rate is like, see if these cells survive that process and they can then be detected with the ICPMS. So this is what these cells look like. So if we start on the left-hand side here, the first image here is of those cells quite happily in a tris buffer. So that's the cells in their sample vials or well plate wells before we've done anything with them. Then we move to the next image over to the right. Now this is an image of the same cells as they pass after they've passed through the sample loop of the MVX 7100 microliter workstation, and then showing them that they're intact still. And then again, the next image, these are the cells after they've passed through the 6-port switching valve. So this is what the cells look like after they've emerged from the automation system. So after they've emerged from the automation system, the next hurdle is the nebulization process and the spray chamber. So what we've shown here, there's two types of images here, so showing the cells after nebulization. So the image at the top of the grouping on the right hand side here, these are cells which have passed through the glassware with a nebulizer gas flow rate of 0.1 liters per minute, showing cells intact, which is exactly what we want. Now the images just below that, these are at a higher nebulizer gas flow rate, showing a different set of conditions, and they show that there are some intact cells as they pass through, but there are also some cells that haven't survived. They've broken down, and they will have broken to the point where contents will have been released, and you will find that a lot of the information that you want, a lot of the signal data, will have fallen into the baseline. Excuse me. But what we've shown here is that up to this point, just before the ICPMS, we've got what we want. We've got cell integrity of this cell line that's gone through, and we've also got a transport efficiencies that we want. As I mentioned before, with this glassware, we're looking at 50, 60, 70% transport efficiency, depending on the cell types that you're using. So next, the ICPMS instrumentation. So as I mentioned, there is the current generation of ICPMS instrumentation that's available on the market is fantastic. You know, we have more sensitivity than we've ever had before available to the analyst in the laboratory. We have got more robust systems, more robust plasmas. We've got more flexible software to handle data. So we're in the best position to really forge ahead with this what we're referring to as this single cell analysis. So more sensitivity allows us to get to low concentration elements that are present in these cells that we might not have been able to see before. Also, what we'll be looking at is quite a broad range of analyte concentrations in the cells that we're interested in. We might be looking at high concentrations of a drug which has been delivered to within a patient, samples have been taken, and we might be looking at high concentrations of something like platinum. We might be looking at cofactors as well, so we might be looking at other analytes of interest which are associated with the primary analytes that we're looking at. They might be a lot lower, they might be present at a lot lower concentration. So that analytical sensitivity that we have with the latest generation of ICPMS is fantastic, and that allows us to do a lot more than we could do before. Now, the analyte suite that we might be interested in, this is where it gets us to the idea of multiple analytes, because what we will likely find is that we're not just interested in one analyte. We might be interested in 2 analytes, or 3 or 4, or even more than that. Certainly in the case of drug studies, where we're looking at the uptake of certain drug molecules and the metal associated with them, if we're looking at that, then we're most likely going to be interested in the other effects of that molecule passing into a cell. So we'll be looking at the concentrations of other elements present. We might be comparing this to something like phosphorus, just to be sure that we're looking at cells as we're passing through. So where we are currently with the latest generation of ICPMS is that we've got more flexibility to be able to look at possibly more than one analyte as these cells pass through in quadrupole-based instruments. And we're also certainly in a position now to be looking at a larger suite of elements with anything time of flight based. And then we go to data processing. So we also have this great opportunity now to look at a lot more data than we ever could before, and in different forms as well. So as I mentioned before, we have to question what kind of data do we want out of this. Are we interested in element data, element concentration data? Are we interested in population data? Are we interested in cell size? These are all possible with the latest generation of ICPMS and their operating software. Are we also interested in other new measures that we've not really discussed before? So analyte per unit something. Are we looking at a combination of concentrations and populations and cell size as well? So there's certainly more to discuss in terms of the data output, but the ICPMS instrumentation is completely open at the other end of this after we've got our cells through the glassware. So just to give you an example of some data output, this is data that's come from the University of Oviedo. Just to give you an example, so a comparison between selenized yeast and regular yeast samples. So these are yeast cells. They're in a certain media, and they've been passed through this process from wells, some well plate wells. They've gone through the MVX system and this glassware interface, and then they've been detected. So we start off with regular yeast on the left-hand side, so the 2 traces here for phosphorus and for selenium. What you can see here is that you can see that cells are passing through according to the phosphorus trace. So this is phosphorus detected as an oxide at master charge 47. So we know that cells are going through here, but you can see below that there's very little selenium coming through as well, because this is just regular yeast. It hasn't been doped with selenium. Now, when the same process is, when different samples are subjected to the same process, so selenized yeast cells in this case, and what you can see here is, again, on the top trace here with the phosphorus, you can see cells passing through that system being very nicely detected there. And then underneath you can see that we've got corresponding with the phosphorus trace above, you've got selenium, which has been detected as, again, as an oxide at 96. So you can see here from this data, we could quite easily take population data, we could quite easily take signal data to turn that into concentration data. So what I'm going to end on in this presentation really is we've arrived at a point whereby we've got a good package for the sample introduction for single cells and the single cell analysis concept in terms of automation and the associated glassware. We're very happy that we've got to a very good point in terms of transport efficiency and cell integrity, and that we can now move on with this package going forward. Obviously, as we go ahead, any improvements that we can make, fantastic. But we're in a good position to start. But what I wanted to end on here is sort of drop in front of what you might consider to be a growing community of analysts and researchers and project managers on single cell. That we are in the infancy of this kind of analysis, much like we were with laser ablation a number of years ago. And there are a number of things to start looking at, and start looking at almost on a community basis. So one of the obvious questions that I've received from a number of people as we've been working on this is, how does one standardize the process in terms of quantifying the concentration of analyte elements in cells of interest? So there's a number of ways that we can approach that. But one question that you might ask is, well, is there a fixed way to do this? Is there an agreed, approved way to do this amongst a group of researchers? Currently, the answer is we've got a lot of approaches that we could take to standardization, but probably not a fixed one that's been agreed on. Also, how does standardization work in terms of different cell sizes and different cell shapes and different cell integrity? What we're looking at here is a very broad type of samples in terms of the different cell lines that you might be interested in. They will all have different structural integrity. At the moment, we are working on the assumption that the cells that we're looking at are spherical in shape. And we work with that assumption and that we move on, but not all cells are spherical in shape. Some of them are more sort of oblong. You can get some cells which are certainly more sort of boxy, but they are different shape. They are different sizes. How does that then correspond to the data that we get out of that, and how does that correspond to quantified analyte in those cells? So there's a number of questions still to answer in this process. So I would add to that, how do we standardize for specific cell sizes and cell shape? So that, I think, is certainly at a community level something to look at and consider. The same could be said for quality control, because that falls under standardization as well. On top of that, I would suggest inter-laboratory comparison as well, between laboratories, between different sites, between different researchers who might have different equipment. They might have different ICPMS systems. Some form of comparison to standardize and improve the quality across the board of analysts, to arrive at a point whereby the data output from a particular laboratory can be related to a certain standardization process. And if we get to that point, then I would suggest that the data output from that could very nicely be used for decision making at that point, certainly in biological problems. And then beyond that, something that we're looking at in addition is the concept of high TDS testing, so which takes us back to the kind of sample matrix for individual cell types. So there will be different buffers that we're working with. We're looking at different sample types, sample media, and where we can go with this. So that is certainly an area of discussion, that an area of further work and research that we're looking at as well. So at that point, I'd like to just make some mention some acknowledgements here. So I'd like to say a big thank you to Agilent Technologies, who have been working with on the single cell process. Of course, Glyn Russell at Glass Expansion, who's done a huge amount of work to bring us to this point of having the glassware interface that allows us to maintain cell integrity through the whole process. Jörg Bettmer at the University of Oviedo in Spain, who's doing some fantastic work with this package of MVX and the glassware, and Dr. Tanaka and Professor Ogre from Chiba University as well, exactly the same, working with the MVX system and the glassware. So thank you to all of you, and all of this work, all of this effort has brought us to this point, which is fantastic. So at that point, I'd like to say thank you for your attention. Any questions, please forward them to us via email. My email address is here. I'm completely open to any questions, and please let me know if you'd like to discuss any of this further. I'd be very, very keen to hear from you. So our webinar schedule through the rest of the year, there are a further 2. There's another webinar in August on the 7th. For the Aridus 3, and there's a further webinar as well in September on the 19th on the AS Express in terms of maintenance and maintaining optimal performance. So thank you very much for your time. Thank you for joining the webinar, and I hope to hear from you soon. Thank you very much.
Okay, thank you, Pete. As Pete mentioned, please feel free to direct all questions to him directly at pete.winship@teledyne.com, and we thank you so much for attending.